and we're going to kick off the fall semester nutrition symposia. And our first speaker will be Dr. Chilton, and we're going to introduce him in a second. Where is our introducer? Oh, here you go. Why don't you take it away?
All right. Howdy, everyone.
Hello.
I'm here to introduce today's speaker, the director of the Center for Botanical Lipids and Inflammatory Disease Prevention at Wake Forest Health Sciences at Wake Forest University Baptist Medical Center. He serves as a full professor in the departments of Physiology, Pharmacology, and Molecular and Translational Medicine. In 1984, he obtained his PhD in biochemistry from Wake Forest University and was given the Cowgill Scholar Award and the Sigma Xi Research Award. He also received Distinguished Academic Achievement Award from Western Carolina in 1999, the Distinguished Service and Teaching Award from the Italian Congress on Allergy and Immunology, and the Denham Harmon Outstanding Research Achievement Award. In 1999, he founded Pilot Therapeutics, and he served as President, CEO, and Chief Technology Officer from 2000 to 2003. Developed a medical food for the amelioration of asthma, allergies, and arthritis called Aerozin. In 2003, he was named Ernst & Young Entrepreneur of the Year for the Carolinas. He is also committed to several nonprofit organizations, serving as founder and president of Heroes Helping Heroes, and is chairman of the Persecution Project that serves the people of Sudan by offering education and aid to victims of civil war, genocide, and religious persecution. As a well-established author, he wrote Inflammation Nation in 2005, Win the War Within in 2006, The Gene Smart Diet in 2009, and currently co-authored Made to Crave the Action Plan with Lisa Turker's New York Times best-selling author. Today he will speak on his recently published review, Diet, Gene Interaction, and PUFA Metabolism, a Potential Contributor to Health Disparities and Human Diseases, in which he will provide us with extensive knowledge of this new expanding branch called nutrigenomics, where genetic modifications are induced by the dietary incorporation of polyunsaturated fatty acids. So let us welcome Dr. Skye Chilton.
Thank you for that wonderful introduction. I just wish my mom had been here. That's really quite nice. Thank you. I really do appreciate it. And thank you for coming today. This is my first time to College Station, and I've been very, very impressed this morning. So thank you so much. Let's see if we can. Is this the one? This is the one. Sorry. The other?
That one. That's this.
Okay. Which one?
Let's go forward. Let's go back.
Okay. Okay, well, now that we've established that, I'm going to talk today about an example of personalized nutrition. Now, that's a word that received a lot of buzz for the last 20 years, but I think today, certainly, I'm going to present what I believe to be a real example, and a real example that has real impact. And I think it's an example that creates huge opportunities. It's huge opportunities for understanding health disparities, why different ancestry populations suffer much worse from the modern Western diet than others, huge opportunities for prevention, as Rob has been hitting me with, but really huge opportunities. And essentially, personalized nutrition is the combination of dietary inputs with humans and their distinct genetic systems, and as you're going to see, ancestral-based genetic systems, giving rise to dramatically different types of metabolism. And we're going to talk about polyunsaturated fatty acids today, but as you can see before, I'm going to have a real emphasis on haplotypes and haplotypes in different regions of the world, and the haplotypes and how those haplotypes really affect the way one of the most common nutrients in our food supply is metabolized. I don't know why that. So, how do gene-diet interactions arise? Well, nothing makes sense except in the light of evolution. So our gene-diet relationships, our relationships with our food and our genes have developed over the last 500 years. But our diets have changed dramatically in the last 50 years. And that dramatic change, as a result of the modern Western diet, has really produced some diet-gene interactions. And these nutrition transitions in calorie and fat ingestion have only emerged in the last 50 years, making maladaptations for that 500,000 years of evolution an inevitable outcome. And the genetic effect of these maladaptations will likely be transmitted not only to this generation, but from generation to generation. So let's think a little bit about at least the human development and timeline of Homo sapiens. So Homo sapiens arose, or are thought to arose, from 180 to 200,000 years ago. So they arose about 180,000 years ago in Central, and I should have Southern Africa. And again, the early diet was a foraging diet, very high fruit, vegetable intake, minimal meat, grain, dairy consumption, honey once you found a honey tree, but that wasn't very often. And again, fiber consumption was about 100 grams a day. Hunting, or at least organized hunting, or hunting in a way that was extremely efficient, occurred about 50,000 years ago. Now, the great expansion from the Central and Southern Africa happened about 80,000 years ago, so that was a point in time in which humanity moved away from the water in the Rift Valley and in Southern Africa. But then 40, 50,000 years ago, humankind moved out of Africa into Europe and began their migration through Asia. Again, occurring at a time when organized fishing really had begun to take place. Organized, or at least fishing hooks, and organized fishing hooks and fishing 12 to 14,000 years ago. This was about the same period of time when humans were beginning to move across the Bering Strait into North and South America. 5 to 10,000 years ago, domestication of plants and animals. Again, the period of time when North and South America was being inhabited. Of course, there was the unfortunate transatlantic slave trade that moved many populations, many African populations, into North America, and the European migrations that moved many populations into North America, thus creating the ultimate almost in admixture populations. Populations of every ancestry, and then the emergence of the modern Western diet in the past 50 years. So what happens when genes and diets collide? Well, what happens is you see populations move from, so you see populations move, and you see the Gaussian distribution move rather dramatically, and we've seen it with diabetes, we see it with a whole range of disease, but you see whole populations and the Gaussian distribution of those populations increase with much higher numbers of cases than you saw before the movement. And so, what are the evidence for diet-gene interactions? Well, this was a study that was published about a year ago in JAMA. And if one looks at what kills us, both in terms of numbers or disability years, certainly at the top of the risk are dietary risk. So the things that are killing us are dietary in nature. Evidence for diet-gene interactions. If one looks, again, at another way at the things that kill us, certainly heart disease, cancer, stroke, these are the things that kill us. Now, fascinating on this is that Alzheimer's disease in 1980 was the 29th leading killer. In this graph, it has moved to number 6. Recent research 3 or 4 months ago suggests it's number 3, and if properly looked at, it killed as many people as cancer last year. So again, these are the things that are killing Americans. And if one looks in different populations, different ancestral-based populations, what one will see is it punishes particular populations much worse than others. So if one looks, for example, at the life expectancy of white females versus black females, white males versus black males, one sees that much lower lifespans. If one looks at the cancers and up here African ancestry populations as opposed to European ancestry populations, one sees much, much higher rates of cancer in these populations. And again, if one looks at obesity, African ancestry populations, Hispanic populations, if one looks at cardiovascular disease, if one looks across the spectrum of obesity and inflammatory diseases, one can see that certain populations are being punished much worse than others. So that's my introduction. That's kind of the setup of the problem. The problem is that we're all dying of diet-related diseases. We're all being punished. We're losing a lot of lives. We're losing a lot of years. Certain populations are losing them at a much worse rate than others. And that's kind of the bottom line from this. So from now on, we're going to talk about fatty acids. So this is a transition, and as we talk about fatty acids, I want to kind of get some common nomenclature. Taking trans fatty acids out of the equation, fats can be divided into saturated fats or unsaturated fats. Unsaturated fats can further be divided into monounsaturated fats. Most well-known are the omega-9 or the N-9 fatty acids. Again, well-known oil for that is olive oil. The polyunsaturated fatty acids, of course, are the omega-3 and the omega-6. Certainly the big one, omega-6, corn oil, safflower oil, sunflower oil. Of course, the omega-3 are the fish oils and the 18-carbon omega-3s that contain alpha-linolenic acid are in flaxseed oil. So I'm going to talk about a set of events that I believe, or at least our research shows, there's a high likelihood are driving health disparities in this country, driving disease in general, but driving health disparities specifically. So in 1961, there was a dramatic change in the amount of omega-6s that went into our diet. And that was driven largely by one decision. The American Heart Association said saturated fatty acids are bad. We're in a food science department. The food science people said, okay, cool. We'll make polyunsaturated fatty acids, so they made cooking oils. And when they made cooking oils, what they did is all of a sudden you see this dramatic increase, and in particular soybean oil, which goes from less than 1% of our calories on a daily basis up to greater than 8% of our calories. So there was an 8-fold increase. It can be argued that there's no place in the modern Western diet that increased so great in the past 40 years as the increase in omega-6s into our food supply due to the addition of soybean oil. Now, once this fatty acid enters into the pathway, it can be elongated and desaturated to arachidonic acid. Arachidonic acid can then form a whole series of eicosanoids. Eicosanoids can then affect a whole range of inflammatory and neoplastic responses. And with all that comes a whole lot of disease. Now, I'm going to give you evidence for every step in this pathway today from our laboratory. So, as I said before, if you're going to produce a diet-gene interaction, one of the first things that typically happens is there's a dramatic change in the human food supply. That happened in 1961, and it was absolutely dramatic. And again, as I said before, it happened because all of a sudden you went from 1% of our energy as soybean oil to 8% of our energy, and soybean oil contains, as you can see, 54% of its total fatty acids are the omega-6 fatty acid linoleic acid. So, dramatic increase at a particular point in time in the modern Western diet. Now, one of the things about being old, there are very few advantages to being old, but one of the advantages to being old is you remember old pathways, and you do enough experiments to you begin to remember data, and if you put enough data together, you see enough things that all of a sudden you begin to want to test. So we began, and I had seen over the years enough data as when I brought in an African American or European American, the African American almost always had double the amount of circulating arachidonic. So again, when we began to think about this, and then we got into, and now we've looked at 3 massive cohorts, and this is one of those first cohorts we looked at. But yes, indeed, when you look in African ancestry populations, African Americans or European Americans, and you look at arachidonate levels, you're going to see almost twice the level of arachidonate, circulating arachidonate levels in African ancestry populations. And this is a p-value you can love, so you can believe that result. Now, in particular, if you look at the whole elongation and desaturation pathway down, you see no difference in linoleic acid. So essentially what this is telling us is there's not a dramatic difference in the ingestion of linoleic acid. And I grew up very poor in the South, and I can tell you in the South and growing up very poor, we eat about as bad as you could eat. So we're not eating any worse than the African ancestry populations. So linoleic acid is the same, but as you move down this pathway, you come to this step between dihomogammalinic acid and arachidonic acid, the FADS1 step, and you'll see lower levels of dihomogammalinic acid and much higher levels of arachidonate. And if one looks at the ratio, and what that suggests is that in human populations, there's a greater efficiency at that step in African ancestry populations. And that's what the data suggested. So we went about testing that hypothesis. Now, any of us, again, great thing about being old is you remember old things, and most of us who've been working on fatty acids all of our life understand that these desaturase steps, the FADS1, unfortunately the geneticists really screwed us up, because that used to be called delta 6 desaturase. And that used to be called delta 5 desaturase. So if you remember them as that, that's okay. But the geneticists call them FADS2 and FADS1. And most of us who have been doing this a long time remember that those are the rate-limiting steps in polyunsaturated fatty acid biosynthesis. So if one looks at those steps, what one will find their genetic location is, they'll find on chromosome 11, they'll find these two genes, or the genes that encode for these enzymes, and they'll see one is transcribed in one direction and one is transcribed in another direction. So on chromosome 11 in the 12-13q region, you see these two genes, you see SNPs throughout these genes, and so we began to look at the SNPs, and in particular we found one that turned out to be very important. We found many, but RS174537, a few years after we began to study it, was shown in a large GWAS study to have a p-value of 10 to the minus 49 for predicting how much arachidonate you can produce. So again, this particular variant was a lot more influential than we thought. We started the work about 5 years ago, and this is a lesson to students. Sometimes it's important to look out in the future and see where the world is going, and see if you're going to be able to compete in that part of the world. And so 5-6 years ago, I determined that my basic biochemistry was going to get my butt kicked unless I become a geneticist. Now, it's not easy to become a geneticist, and I'm still not a geneticist, but all of a sudden, my colleagues at Hopkins, where I was for 6 years, I went up there and said, please teach me genetics. So my lab took a dramatic shift 6 years ago when we began to understand genomics and genetics. And we began by looking at an island that Raska Mathias at Hopkins and I had looked at when I was at Hopkins. And it's the Tangiers Island off the coast of Maryland. And it was a great island to study because it was genetically homogeneous. And it only had one boat that came on the island every day, and so everyone eat the same thing. So you basically have a genetically homogeneous population and you're controlling diet. And you have about 600 people on the island. So we began to look at this, and yes, indeed, we began to look in the FADS cluster, the genes that I've told you at 11, 12, 13q. And what we began to see were these very, very significant, and again, this is a large LD block, and I'll come to that in a second. But these SNPs in the FADS cluster very strongly, and forgive me, anytime I see a slide like this, I say, screw that person. But this is really, if you just look at the arachidonate, and if you look at the arachidonate to DHA ratio, what you'll see in this population are these SNPs in the FADS cluster that do an extraordinary job at predicting whether or not you make arachidonate from linoleic acid. And again, just to show the data in a different way, if one looks at arachidonate, and this is RS174537, if you're GG, you produce a lot higher amount of arachidonate. If you're TT, a lot lower amount. And in this Caucasian population, the frequency of TT is much lower than the frequency of GG, and the heterozygotes and homozygous are about the same. Again, if one looks at DHA, you see it just flipped, exactly flipped. DHA is the precursor to arachidonate. Again, suggesting that that enzymatic step was working much more efficiently. And again, if you look at the arachidonate to DGLA ratio, you see a much higher level in the GG versus the TT variant. So we then, at that point, began to wonder if that was responsible for us seeing much higher levels of arachidonate in African ancestry populations. So we began to look at that, and we began to look in several cohorts, but we looked in this particular, and I forget, I think this is Gene Star at Hopkins, but we looked in several cohorts. And basically, if one looks at arachidonate, and we'll just focus on arachidonate, whether you're African American or whether you're Caucasian, if you have the GG variant, you're making much higher levels of arachidonate from linoleic acid. If you're the TT variant, much lower levels. Now, you can tell from this if you only see these two TTs down here, and believe me, we had to oversample like crazy in these African ancestry populations to find two TTs. You can see a dramatic different frequency. Now, I'm going to show you that, but you're going to see a lot more GGs in the African ancestry populations than TTs. Now, not only do you see these genotypic differences, but if you look within the genotypes, there's some genetics that we've not accounted for, and we've still not accounted for that. But what these data suggest is there is a 50 to 60% genetic component that determines whether linoleic acid goes to arachidonic acid between African and European ancestry populations. About 30% from the FADS variant at RS174537. Now, most of you, and I should have pointed this out in the early biochemistry pathway, but you also have an omega-3 pathway that shares the same enzymes as the omega-6. So we also address the question, are you seeing similar differences, and this is arachidonate to LA or arachidonate to DGLA, it's just two different cohorts, and this is, but this is the omega-3, and what one sees is African ancestry populations also make more omega-3s. So going down that pathway on both sides, these FADS desaturase variants are rendering these populations better able to produce long-chain polyunsaturated fatty acids. So we then went to the Human Genome Diversity Panel, and as we had suspected, what we saw is if you look in African ancestry populations, what you see in Africa is almost 100% GG, the efficient variant. Surprisingly, in Native American ancestry populations, almost 100% the non-converted. And then admixtures between, you see everything. And this just happens to be from 3,000 or 4,000 people that we've studied in Piedmont, North Carolina. But in Piedmont, North Carolina, if you're an admixture, if you're an African American in Piedmont, North Carolina, 81% of African Americans are GG at RS174537. 40%, only 47% of European Americans are GG at RS174537. So again, strong differences in the genotypic frequencies that make arachidonic acid. So I'm showing you the pathway from another perspective. So in this particular perspective, you see the dramatic increase in linoleic acid next to alpha-linolenic acid as a result of adding cooking oil to the modern Western diet. Now, I've shown you the FAD steps are highly controlled and rate limiting. Are there other steps in this biochemical pathway that are important? Because there's several elongase steps, there are several beta oxidation steps. So to address that issue, or to address that question, we looked at the MESA cohort, and we looked at 7,000 people from the MESA cohort, and we took SNPs throughout. We got 90% coverage of all of these genes, and we looked throughout the pathway to see if is it just the desaturase steps or is it other steps. And this is the desaturase steps, and the the significance is much higher in European Americans for one reason. It's because they have 3 genotypes, and African ancestry populations only have the GG and the GT. So it's much easier to get significance here, but again, throughout the FADS cluster, and you don't have to look at all of this, but whether it be omega-6 or omega-3, FADS cluster variants are they have dramatic effects on levels of these long-chain PUFAs. If one looks at the elongation steps, EOVL5 or EOVL2, one doesn't see a whole lot. You don't even have to look at it, you just don't see red. If one looks at the beta oxidation steps in the pathway, and to be quite frank, Howard Sprecher, a good friend, was a good friend, and he worked out the Sprecher pathways utilizing these beta oxidation steps, and quite honestly, we still don't know that pathway completely, the biochemistry of that pathway. But here are a couple of genes that I suspect may be playing a role, and so we looked in those genes, and again, no effects. So once again, what the data indicated was FADS cluster gene variants, very important in determining whether or not you can make arachidonate or long-chain omega-3s, and much higher frequency of efficiency converting variants in African ancestry populations. Well, as the other camp, and we're in the midst of a debate with the American Heart Association, and I call them the other camp. As the other camp would say, so what? So what? Arachidonate's not bad for you, so because it just circulates and it really has no effects on icosanoids. So we, in a recent JVC paper, addressed this question, and basically, it's not a simple question because, again, the great thing about being old is you remember all this biochemistry. So for arachidonate to get to icosanoids, it has to go through a whole lot of biochemical steps. And then it goes through the acyltransferase steps, CoA-dependent acyltransferase, the CoA-dependent synthetase. It goes through the CoA-independent transacylases. We actually discovered this enzyme before when I was a little boy. And so you go around the pathway, but to get to free arachidonate is a very complex pathway. So the question is, do FADS variants in this part of the pathway impact this part of the pathway? And to do this, we collaborated with my mentor, Bob Murphy. Keep pulling him out of retirement and keeps kicking my butt, but he's the best in the world at LC-MS, MS, and with icosanoids discovered leukotrienes, and so we looked and we simply did whole blood stimulation. So Zymazan whole blood stimulation. Before you stimulate it, you see all these internal standards. And after you stimulate it, you see a 5-lipoxygenase response. So I can't really speak to the cyclooxygenase part of the pathway, but in this part of the pathway, you get 5 heat and leukotriene before you get the 15 heats as well. And basically, what we were able to show is at RS174537, we of course got what we would predict. You make more arachidonate if you're GG versus GT. But again, we got this very strong association with your capacity to produce the pro-inflammatory LTB4. So again, it does matter. Variants in the FADS cluster work their way down to icosanoid generation and the capacity of whole blood to produce icosanoids. So what about the rest of the pathway? Again, the other camp is fighting, so what, so what? You make more icosanoids, that doesn't mean you have more disease. Well, we can't answer that yet, but what we can show, and this is from a very recent paper that we published, and I apologize for this paper, this figure, because it's one of these, oh my gosh. But what I do want to show, and what I want to point out is, first of all, you see the FADS cluster right here in B, and you see FADS 2 here, FADS 1 here. You see the LD blocks in African or African Americans or European Americans. You see these LD blocks. And then you see all of these SNPs. And what we've done is we've listed the SNPs in here that have been associated in a whole variety of either GWASs or looking at individual or SNPs. And basically what you see is there is a dramatic association between FADS, SNPs, the ability to make triglycerides, cholesterol, and as well as coronary artery disease. So again, you get all the way to the end. SNPs in the FADS cluster are associated with higher arachidonate, higher eicosanoids, all the way out to coronary artery disease. So once you increase your amount of linoleic acid in your diet from 1.5% of calories to 8% of calories, and you have variants that much more efficiently convert that to arachidonate, and those variants are then associated with cholesterol, triglycerides, LDL cholesterol, and coronary artery disease, you're beginning to make a compelling case that diet-gene interactions can drive both disease and health disparities. So, one of the arguments the other camp has made is it really doesn't matter that you feed all this linoleic acid, because all of this linoleic acid, only 2 to 3% can get down this pathway because of, and I like to think of them as plumbing pipes just because I'm very simple, but these FADS, rate limiting steps, only 2 to 3% can get down the pipe. And so if you have 8%, it really doesn't matter because only 2 to 3% are getting down the pipe. Well, certainly our data suggests that if you're GG or if you're African American with a very high likelihood of being GG, which is greater than 80% in North Carolina and I'm sure everywhere in the country, then the amount of energy that from linoleic acid that comes down this pathway is likely to be much, much larger. So, if you'll remember the slide where this appeared to happen in Africa, I'm going to talk about a study we just published, and this is something as students you never want to do because you'll get your butt handed to you, but I'm going to do it anyway because this study was done largely by bioinformatics people in Seattle, in particular Josh Askey utilizing Bill Gates' supercomputer, and I can't tell you just how the data arose, but what the data showed was this particular mutation arose 85,000 years ago, or approximately 85,000 years ago in Africa. This GG, so G was the derived allele, T was the allele before, G was the derived allele, and this mutation took place 85,000 years ago on the African savanna. Now that's an interesting period of time because that's the period of the Great Expansion. So the period of the Great Expansion is, it's felt by many of us that humankind lived around the water, the Rift Valley, and in southern Africa, primarily because of the need for DHA for these huge brains that Homo sapiens had in the huge complexity of these brains. And leaving the water would have meant that there would be no source of DHA from third trimester through second year of life for baby. And that would have been devastating. So if humans did not have the ability to take plant-based fatty acids and convert them to long-chain PUFAs, then they would be tethered to the water. And they did not have that capacity before these G variants. So I'm sorry.
Just to clarify then, this is fixing the GG allele. This is where the GG allele comes in.
This is where the GG allele, this is where the G allele comes in. And just to kind of show you the data, and there's I've got but what you can see and what appears to have happened is 85,000 years ago, as best as that computer can fix it, there were this G allele derived on the African continent and went to fixation.
So why are these compounds more available in the water than they are on the land? How would you get them in the water?
From shellfish, DHA from fish. So to be around the water, the only way you can get long chain PUFAs is from fish or from shellfish. So it's envisioned, not by me, by people a lot smarter than me for the last 50 years. This is not my hypothesis. But the G allele derived and the G allele went to fixation. And what we show here is the selective pressures that were on in that particular region. And these are some of the highest selective pressures we've ever seen. So these selective pressures are in the 99.5% selective pressures that have ever been measured. So this was a very important evolutionary event. However, and I'm going to move to the next, what's the molecular mechanism of action? How do these SNPs impact polyunsaturated fatty acid levels? Now, again, the most obvious way would be transcription. So if you have certain of these variants, you're going to make more of the enzyme, and if you make more of the enzyme, you're going to be able to convert more of linoleic acid or alpha-linolenic acid to arachidonic acid or DHA. So we began to look at the molecular mechanism of action, and in particular, RS174537 sits out here. Now, I don't know much about transcription, but it just is not sitting in a place that it wasn't sitting in a place that looked like it had a lot of regulatory capacity from a lot of perspectives. So, and I don't know why one night I woke up and said, well, I bet it may be tracking a methylation site. But I did, and we had 144 liver autopsy samples, and we had performed 345,000 methylation scans, looking at 345,000 methylation sites, and we began to look at the association of this with methylation throughout the FADS cluster or throughout the human genome, as far as that's concerned. So the idea is, and we've seen now probably 10 examples of this in the literature, and we don't know how it happens, but it's called allele-specific methylation. And there's an allele, and somehow it affects the methylation, the epigenetic regulation, or the methylation of CPG sites throughout the genome, and it can do it at different distances. So we tested that hypothesis in the PDAY study, and basically we took 77 Caucasian, 77 African American subjects, the livers from them. We carried out SNP genotyping, and then I said 385,000, I meant 485,000. What's 100,000 epigenetic sites? But in any event, we looked, we did full GWASs. We also looked at 485,000 methylation sites throughout the human genome. And sometimes it's better to be lucky than good, and we were certainly something. Because what we found was RS174537 only lit up a few methylation sites, and they sit right in the FADS cluster. Now, again, they sit in the FADS cluster, and this is a Manhattan plot, and these are chromosomes, and again, this is a p-value of 10 to the minus 29, so we could believe the data that RS174537 and the genotype at RS174537 was linked to that. Well, when we took a closer look at where it was, and I'm going to have to speed up because I've got 10 more minutes and I need to go, but what you see is you see this, and if you look to see where it's located, it's actually located between FADS1 and FADS2. Now, that's a fascinating location because FADS1 is transcribed in this direction, FADS2 in this direction. We then looked in the ENCODE data, and this particular epigenetic site, this particular methylation site, is sitting right in the middle of an enhancer. So again, that was fascinating because enhancers bring together the three-dimensional structure of promoters and genes to allow them to be transcribed. So whether this was methylated or not, we propose determines whether or not these genes are transcribed. And just to kind of show that these are genotypic, so this is GG, GT, and TT in African American, or I'm sorry, this is FADS1. And basically what you see here is that RS174537 very well predicts whether or not that site's going to be methylated, but they're outliers. So even if you look here, you can go from 25% being methylated to 75% within the same genotype. Now, we think that that's due to epigenetic changes, whether or not that person has been exposed. The other exposure, that this methylation site is a much better marker of fatty acid biosynthesis than the genotypes themselves, but that has yet to be tested. In any event, this was very exciting because outside of the inactivation of the X chromosome, the largest degree of allele-specific methylation that had ever been seen was 6 to 7%. Well, you can see that we're seeing 50 to 60% changes in methylation based on genotype here. So again, we believe that this enhancer and the methylation of this enhancer, and we're looking, can play a critical role in whether or not these two genes are transcribed. So again, the idea is PUFA exposure, age, sex, pregnancy status, a whole variety we propose can impact the methylation status of this. Certainly, somehow, by ways we don't understand, RS174537 and other SNPs appear to be able to sense that methylation site. We are proposing it controls FADS1, FADS2 activity, and we're just in the middle of all this. Again, with all the things that come with that, including the fatty acids themselves, the biomarkers of human disease, and the diseases themselves. So what are the concerns that arise from these data? Well, the American Heart Association says that 5 to 10% of our calories should be linoleic acid. That's, we believe, problematic. If we're telling, specifically they say 5 to 10% of your calories should be PUFAs, but in the context of the modern Western diet, there is no ALA, or there's very little ALA unless you eat flaxseed oil, so that means linoleic acid. So the current recommendations were based on registered clinical trials. I think there were 41 of them carried out in Caucasian populations. So we think that that certainly gives reason to have pause. Even within a Caucasian population, and then this is a recent study, Joe Hiblin and Chris Ramstead, my buddies at NIH, are just kicking the American Heart Association's butt over this right now. But this is a reanalysis of the Sydney study, and basically what they did in this reanalysis of the Sydney study is this was taking linoleic acid or taking soybean oil up to 15% of calories, as opposed to saturated fatty acids, 15% of calories. And this was humans, and this was cardiovascular or cumulative deaths or cardiovascular deaths. And you can see that the polyunsaturated fatty acid people died a lot more readily. One of the confusions in this field is most of the studies that were done for the American Heart Association recommendation were done by giving mixtures of omega-6 and omega-3s. And when Chris has looked at this further, and when he has looked at the linoleic acid selective trials versus those that mix omega-3 and omega-6, he sees that the omega-6 trials in this meta-analysis cause heart disease. The combination has some protective effect. We have a great deal of concern, especially in certain populations, especially during pregnancy. Again, from 29 weeks through 2 years of age, the brain explodes in long-chain omega-3 polyunsaturated fatty acids. And if one looks at RS174537, and if one looks at the genotypic frequencies, one will see, and I'm particularly concerned in native ancestry populations, where the vast majority of individuals are TT. So the question is, with the TT genotype and no omega-3s in the diet, where does mom get omega-3s? And it's really an important question. You know, right now we're looking for reasons for this explosion in a whole bunch of developmental disorders, because they came out of nowhere and they have absolutely exploded. ADHD and autism. Now there's a recent meta-analysis in ADHD, either looking at peripheral blood or looking at postmortem brain tissue, and there are marked decreases in omega-3s in the brains of these children and the periphery of these children. So are we seeing gene-diet interactions, and is that the missing environmental factor that people are looking for that are driving these developmental disorders? We lead the world in mental disorders. Why? I don't know, but and some of it may be diagnosis, but in that particular, I heard several people say that in the audience, but in that particular study that was carried out by the World Health Organization, that was controlled as much as you could control. Well, the one that you can't deny is the dramatic increase in Alzheimer's disease. And that is a real deal. And that's the real deal, and we've gone from it being the 28th leading killer. If you believe the recent report that if all cases were called correctly, it is now number 3. It really is. And again, post-mortem fatty acid compositions see, again, dramatic alterations in omega-3 to omega-6 glucose. I've got one more slide. Schizophrenia, again, you see in schizophrenia, you see a subsegment of this population that has dramatically altered omega-6 and omega-3 polyunsaturated fatty acids. This was a post-traumatic stress disorder study that was done by my buddies up at NIH, and basically what they showed is if you had low levels of omega-3s, you were 3 times more likely to take your life exposed to the same levels of combat. So I'm worried about the extremes. I'm worried about the extremes that diet-gene interactions produce. NIH has been very good to me over the last 30 years, and in particular the last 10 years as part of a botanical center. We've been very, very fortunate to have their funding, and my lab, in particular, Raska Mathias at Hopkins. I'm no geneticist, but if anyone could try to turn me into one in 5 years, she has. Susan Sargent runs my lab. These two women complete me. But they're a great bunch, and I'm incredibly indebted to them for the work. Thank you.
Time for a few questions.
So this is wonderful and fascinating, and thank you so much for talking about it. I enjoyed every second. So I want to talk a little bit about Alzheimer's, because you know the interesting thing about Alzheimer's is there's an overlay of sex differentiation. So the incidence of Alzheimer's, again, depending on how you call it and how it's diagnosed, which is very confusing to those of us who are, you mentioned of age. Myself, I was taught back in the day that Alzheimer's was pre-senile dementia. So 85-year-olds didn't actually have Alzheimer's, right? So I don't know where the sense, but anyway, Alzheimer's, for however it's called, is there's more than half, about 2/3 of the sufferers of Alzheimer's are women. And I'm wondering if there's some overlay or interaction. It seems obvious, but you would know.
You know, from our pathway perspective, I'm so happy that we found this epigenetic site, but these CPG sites in this enhancer, because it does appear to be playing a key role. Well, what that indicates is that we can much more, we're not just looking at a SNP now, but we're looking at a range from 0 to 100, and women have gone through pregnancy, and obviously during pregnancy, there has to be a dramatic increase in their capacity to produce them. There doesn't have to be, but there's shown to be. So how does that come about? Well, my first candidate will be, to look right in that enhancer to see if those methylation sites are responsible for that. But do we have ghosting of that later on, and you're taking all that linoleic acid? Because the thing about Alzheimer's is Alzheimer's use, from a fatty acid perspective, if you look in the cerebral spinal fluid, what you'll see is these incredibly high numbers of oxidized derivatives of arachidonic acid. And in fact, the new gene test that just got the gene test that got so much press 4 or 5 months ago were all oxidized phospholipid derivatives of fatty acids, phospholipids. So we'll see.
Pregnancy is a risk factor then?
I don't know.
By this argument,
this argument, it would be. But again, I'm 10 years down the road on the research, and we're not there. We're a long way.
Yes. Go ahead.
You mentioned that there's some SNP variants. The SNP variant that's in the African American population does have some populations that show a low percentage of arachidonic acid, 25% you said compared to some 75%. And then you mentioned fetal reprogramming. So do you think there's possibility for pharmacological intervention of, let's say, high entry fluid during fetal development that might actually end up reducing the arachidonic levels later on? So that.
Yeah. Well, the big concern I have in fetal programming is DHA. Right now, I'm working with 3 different organizations trying to get into Native American populations because yeah, absolutely. I'm absolutely frightened for that population because, 85% of Native Americans are TT at RS174537. It just flips itself from the African American. So my real concern there is a gene-diet interaction with the inability to get enough DHA during the second. And we just completed a study in non-human primates that we're writing up right now, that there's this second major burst of DHA in adolescence between kind of middle childhood up into late adolescence. So it's not only from second trimester to 1 year of life, but there's a second burst we've shown in non-human primates that take place from middle childhood, and that then begins to address the whole ADHD and autism, because DHA turns itself over every 2.5 years. So you can't go back to all the developmental arguments from early childhood. It would be hard to, but some interesting questions that have to be
We have room
addressed.
for just time for one more question.
So the way you draw this is that you take your omega-6, make them down to arachidonic acid. Of course, it is complicated, right? It seems to me, have you ever actually looked to see what the PC component is? Because let me offer an alternative. One is that in certain populations you have higher basal rates of inflammation. That's what gives you your circulating arachidonic acid, all right? And where does that basal level of inflammation come from? Let's get back to the oxidized lipids then. So has anyone actually looked how you deconvolute oxidized omega-6 versus oxidized omega-3? One could actually give you an end product that's toxic as hell, basically. The